Okay. Well, we're going to try and shoot for 50 minutes, so we still have 10 minutes at the end, and then if people want to stick around past that, they can. I have a class at 3 too, so. Okay, so I do have it recording now. There we go.
Is that clicker?
I don't know. Is that yours, Kamal? Okay. Plug it in.
A way to turn off the sound with people entering and leaving.
That was, yes. Let me.
That was. Yeah, no.
Yeah.
That's
I think it's under. There we go.
Nice. Hey. Somebody said, oh, I don't know actually which physics class it was. Somebody said they were getting extra credit. Okay.
Oh, well, for mine, if they write a report, they can.
They write a report.
Yeah. Well, it's 2 o'clock. Should we go ahead and get started then?
Yeah.
All right. Well, welcome everyone who is here in person or attending by Zoom. This is actually our 2nd annual Nobel Prize Symposium. I organized this last year too, and for 2 reasons. I think it's really interesting to hear about the science that makes the headline from our scientists here at Oregon Tech. But I also like seeing how excited people get about the different prizes. So last year when genetic switches were one of the prizes, Dr. Usher was almost like a kid in the candy store, like, me, I want to do that one. And Travis was very excited about CRISPR this week. So I just think it's a good opportunity to learn about the front page science from our own faculty here and just see the connections to what you're learning to a bigger picture. This year's prizes cover a wide range of topics. So we have research on the hepatitis C virus that Dr. Gandhi will be telling us about. Dr. Lund is going to be telling us about CRISPR, the gene editing technique that's been growing a lot recently. And in physics, the discoveries this year had to do with black holes. So a long-standing prediction of physics that is being recognized with a prize this year. Since we're trying to cover a lot of science in a short time, I will let Dr. Gandhi get started. But we've tried to make our presentation so there'll be a little time for questions at the end. I don't mind if you ask along the way. I'll let the other speakers tell you their preferences on that. But turn it over to Professor Gandhi.
Thank you. Afternoon, everybody, both here and online. Let's see, hopefully Zoom will, okay, there it goes. So I'm going to be talking about our first of the 3 Nobel Prizes, one in medicine or physiology. And given that our other 2 are on black holes and CRISPR, which are still relevant in the news today, I thought, the least sexy of them is the discovery of HCV, as this is kind of a complete story. But the 2020 Nobel Prize in Medicine or Physiology was awarded to Harvey J. Alter, Michael Houghton, and then Charles M. Rice for the discovery of hepatitis C virus. And as you will see, the reason that they are sharing the discovery and sharing the Nobel Prize essentially comes down to the fact that each of their labs was responsible for a small part in what eventually led to the discovery of hepatitis C. So just a little bit of background information for all of you. Hepatitis is a term that simply means inflammation of the liver by itself. But while it is a general classification for that disease, it is also a class of viruses. And I'll touch on that a little bit at the end, how these names kind of can be somewhat misleading. But the name of a group of viruses that infect the liver. And so the 3 most common ones that most people have heard of are hepatitis A, hepatitis B, and hepatitis C. Since the discovery of C, hepatitis D and E have actually also been discovered. Hepatitis A is a fairly common virus, but it's usually a short-term acute virus that brings on an infection and then is usually cleared. Hepatitis B and C are both chronic bloodborne infections. Hepatitis B, there is currently a vaccine for, and as you'll see, part of the significance of the discovery of hepatitis C is the ability to now treat it because we know how the disease works. So hepatitis C is actually a member of a viral family called flaviviruses, which includes several hemorrhagic fever viruses and then encephalitis viruses. So it's actually very different in terms of the disease it causes from some of its family members. It is a small plus sense single-stranded RNA virus, which one of the unique aspects of viruses is the fact that they can use things other than DNA as part of their genome. We won't really get into the significance of that in this talk, but it is actually a very important aspect of viruses and how they change. And I'll touch on that a little bit at the end when I talk about some of the important lessons that we've learned from this study. It is also a fairly small virus in terms of its overall size. So it's an envelope virus of about 55 to 65 nanometers. Again, for those of you who are familiar with these kinds of size, you can see how small that is. But if you're not, you can think of a typical person as on average close to about 2 meters. The cell is typically about on the order of microns, so about 10 microns. A typical bacteria is about 1 micron or 1 micrometer. And so another level down from that, about 100 times smaller than even a typical bacterial cell, is how big you would find hepatitis C. This slide shows you what the particle looks like, both in terms of its actual makeup. So it's kind of this icosahedron structure, which is one of the common structures you see with viruses. They somewhat of a soccer ball type shape. You also see the genome down below and the number of genes. So here again, a unique aspect of many viruses is that rather than having a sufficient genome to actually allow for their own growth, they are simply a minimal set of genes that allows the virus to take over a host cell and to grow. And again, we'll touch on the significance of that in just a second. In terms of its disease, hepatitis C is actually quite a major cause of disease around the world. In the United States, there are currently an estimated 50,000 new cases a year. Many of those go unreported because, as you'll see on the next slide, the stages of disease start from acute and eventually become chronic. But it is thought that currently about 2.5 million people are living with persistent or chronic cases of hepatitis C here in the US, with approximately 70 million cases worldwide. And so it is a very significant cause of disease. As someone gets infected, about 15% of people who get infected develop an acute viremia, so they actually notice that they are sick early on, and then they typically recover from that. But the issue with hepatitis C is the chronic infections that occur for potentially a long time after. So about 70% of people will develop an asymptomatic chronic infection, which eventually leads to liver failure or liver disease. And about 15% of people develop severe cirrhosis rather rapidly. And hepatitis C can eventually lead to liver carcinoma. So in 2018, roughly 15,000 or a little over 15,000 deaths were reported here in the US attributed to hepatitis C. And that is probably an underestimate given that many people are asymptomatic, with almost 400,000 global deaths attributed to hepatitis C. So our story for our Nobel Prize then starts slightly before the time of our three scientists, where the first ever hepatitis was discovered roughly in 1945, but it wasn't actually discovered as a virus. It was simply discovered as an infectious disease. The actual particle was not identified until the 1970s. So hepatitis A was discovered officially in 1945, although again, not as a particle until the 70s. And then in the 1960s, a scientist identified a non-hepatitis A virus that was causing hepatitis-type diseases or liver-type diseases. That virus, hepatitis B, was then awarded the Nobel Prize in the 1970s. But a scientist, our first of our three scientists, Harvey J. Alter, was working at the NIH at the time that hepatitis B was discovered, and using the current techniques, he was studying transfusion-associated liver damage, and noticed that quite a few of his recipients that ended up with liver damage did not actually get damage from the current two known viruses, hepatitis A or hepatitis B. And so he presumed that there must be some kind of other infectious disease, and he was able to show that the agent that was causing hepatitis in these transfusion patients was an infectious agent, and that it followed the characteristics of a virus, and that could actually be transmitted in chimpanzees. So he developed the first animal model to actually study what was called non-A, non-B hepatitis. And as I'll talk about in just a second, the development of that animal model is actually very important for how future studies came about. The second of our two Nobel Prize awardees was then following up on those studies looking at non-A, non-B hepatitis. And essentially at the time, there was very limited resources or very limited ways to actually isolate the virus. And so using traditional methods of the time, he was unable to actually isolate the virus that was, nobody had been able to isolate the virus that caused the non-A, non-B viral disease. So his lab actually developed a technique that is pretty cool. Essentially, they took isolates of genetic material from infected chimpanzees, cloned them into bacteria, and let the bacteria make the proteins that were cloned from those DNA fragments. And they found that there was one isolate that they had taken where if they then activated human, essentially human patients that had antibodies against the hepatitis infection, they found one clone that actually caused a protein that was made that was responsible for that antibody response. And so they were essentially able to isolate the genetic material that corresponded to this non-A, non-B hepatitis, which they then called hepatitis C. And based on what they could determine of the genome at the time, they noticed it was very similar to a known family of viruses, flaviviruses, which again include things like dengue and West Nile and Japanese encephalitis virus. Finally, our third of our three Nobel Prize winners then took that cloned cDNA or cloned DNA and was able to then fully fragment and fully isolate the actual genetic material of the virus, which is actually a segment of RNA, and was able to show that simply that segment of RNA, when put into a chimpanzee or into a cell, could then recreate the infection caused by non-A, non-B hepatitis. And so essentially, this was the final piece of proof that was required to show that there was, in fact, a virus that was causing this kind of infection. And so between the three studies, we essentially then have a 25-year course of methodical research that goes into proving that the agent causing hepatitis or non-A, non-B hepatitis is in fact hepatitis C. And so again, initially we have to isolate the transfusion-caused non-A, non-B hepatitis. We then take the DNA from infected patients and use antibodies from patients to then identify a cloned DNA fragment that was actually producing the proteins of the virus. And finally, we actually are able to then isolate the genetic material, which in this case was RNA, put that into an animal and show that it actually produced the hepatitis viral particles. So it's actually quite a long story, but essentially the discovery part of the story is somewhat done. However, I do want to talk briefly about the significance and kind of the what we can learn from the study. So in terms of significance, first off, it simply led to the ability to actually test for hepatitis C in transfusion blood. And so the blood-borne or the transfusion-borne transmission of hepatitis C has almost been completely eliminated thanks to these studies. Furthermore, now that we know the actual genetic makeup of this virus and how the virus transmits itself, we can actually look for antiviral targets that protect people who have the virus. And one thing that I can't really get into in detail, but it is very hard to develop antivirals relative to antibiotics. And so the isolation of that genome was actually very important for that process. And so thanks to this work, chronic hepatitis can actually be now cured with a number of drugs that target hepatitis C directly. And so the CDC actually currently recommends that everyone over 18 get tested for hepatitis C at least once. And if they are in a high-risk population, that they get tested regularly. Finally, I then wanted to touch on some take-homes. Since this is relatively a finished story in terms of the discovery, as opposed to some of our other two talks that you'll hear today, one of the things I wanted to point out about this is that this study is actually a series of experiments that took roughly 25 years from the time that we first noticed that there was some kind of non-A, non-B hepatitis to the time we actually isolated the virus. And as we are currently faced with this pandemic, as new information is coming out all the time about this virus, one important idea to keep in mind about that is that the actual molecular studies of viruses takes a long time. It is really difficult to explain everything that we learn, and it takes time to actually identify and understand all the things that we're learning about different kinds of patients. And so don't expect science to kind of answer things right away, in essence. Second, many scientific conventions were developed long before our understanding reached a cellular or genetic level. And one of the things that, just to point out there, is that when we say hepatitis A, B, C, D, and E, the nomenclature there, it makes it sound like those things are all related to each other. But in fact, all 5 of those viruses are very different in terms of their genetic makeup and their family makeup and how they actually infect the liver. The only commonality is that they have a tissue tropism specifically for the liver, and so they cause liver disease. But that is actually true in microbiology in many ways, simply because for a long time we did not have the ability to study things at a genetic level or at a cellular level. And so lots of naming conventions for bacteria don't really fit what those bacteria are or do. And then finally, working with viruses poses many additional challenges relative to working with any living thing, because a virus has to infect a host, and you can only study it in context of that host. And so if there is no lab system or animal system that you can use to study a given virus, these studies that we have for that virus often progress very slowly until someone is able to develop that kind of animal model or study it in a lab setting in some way. And so that is our, again, our discovery of hepatitis C with our 3 individuals there. I guess we have a minute or 2 for questions, if there are any. Otherwise, yep.
So about when was Dr. Rice finishing up the end of the chain?
So this occurred in around, I think his article was published in the early 90s.
Okay.
Yep.
Could you talk a little bit more about the treatment for Hep C?
So there are a couple different drugs that are given. I actually don't know much about them at the molecular level, but essentially, if you look at the mechanism of infection, there are thought to be a couple of individual enzymes that are targetable by the various drugs. So I can look into the drugs a little bit more, but I'm not too familiar with them offhand, right?
Yeah. So you said you could introduce some of the virus genetic material into a bacterium and it would make some of the virus protein.
Correct.
So what is specific to humans, right? This isn't, hepatitis C isn't a phage that runs rampant through bacteria.
So the process of doing that involves taking the genetic material that you're interested in and putting it into a bacterial plasmid that the bacteria will then take up and express. So the process of essentially making a cDNA clone or clone library involves taking a whole bunch of different genetic markers or just different regions of a genome, putting them into bacteria, and then as long as the protein that's produced is not toxic to the bacteria, and you have the correct markers on the plasmid that will express genes in that bacteria, the bacteria will make a protein because DNA works the same across all cells.
Okay. I thought you were putting the RNA from the virus in there, but that makes a lot.
No, in this case, they had to put the cDNA clone. So they. Yep.
I was wondering, the main channel for this disease is through blood transfusion?
Yes.
I'm wondering why there are so many schizophrenic cases currently in the US every year. Is it because the blood was not monitored?
So at this point, the most common cause of transmission is actually through shared needles and drug usage. So luckily, the numbers have gone down significantly because we have that ability to look for transfusion blood, but that doesn't necessarily stop all activities that would transmit bloodborne diseases. Anything else? Thank you very much.
Dr. Gandhi.
So the next topic is chemistry, kind of. You know, chemists might be a little bit grumpy that there's so much biology winning all these Nobel Prizes, but sort of biochemistry, microbiology, genetics sort of topic here. So The prize was for, is there a pointer on this? Yeah, there we go. Should probably stand by the camera though, huh?
Follows you.
What's that?
It follows you.
Oh, even over there?
Yeah.
Right on. So these guys won the 2020 Nobel Prize in Chemistry for developing a method for genome editing. And the story here, like Kamal was alluding to, is a little different than the Hep C story. So, you know, CRISPR is very much definitely in the news, right? So we're using it to treat diseases in the last few years, or starting to do trials at least. Using it to detect coronavirus. We're seeing news of engineered babies. Could it be the next virus killer? You know, the headlines can be, you know, like CRISPR weapon is spread by bacterial sex to destroy deadly superbugs, kind of like, you know, a lot of hype in the headlines. Can CRISPR save the world's chocolate? A couple of years ago, there was a lot of headlines about how chocolate was going to be extinct by 2050 if CRISPR didn't save the day. This is real. So anyway, CRISPR is definitely in the news at the moment. So first of all, what is meant by developing a method for genome editing? So when we talk about genome editing, what they're talking about is making particular changes to DNA sequences within an organism's whole genome, all of its DNA. So going in and specifically and carefully making edits as you desire. It might just be deleting things or knocking things out, so to speak, but it also might include sort of inserting something that you want, a sequence that you want in there, or replacing some sequences with new sequences. You know, and CRISPR is not, CRISPR/Cas, CRISPR-Cas9 is not the first technique to do this. So there's zinc finger nucleases and talins and things that people have been working on for a long time. But CRISPR really improved, you know, sort of how these could be done. So the previous techniques, they were all right, you know, they're getting the job done, but they're kind of cumbersome and really complicated to do, time consuming and expensive and such. So the real reason that CRISPR exploded the way it did is it's really powerful, pretty simple, really flexible, pretty cheap. So with that intro, we'll kind of delve back into the story a little bit. So there's really kind of just the two characters in this Nobel Prize. Emmanuel Charpentier was born in France, did work in New York, Vienna, all around Europe, and was basically a microbiologist, geneticist, a little bit of a biochemist perhaps. But she sort of starts the story. So in all of her work, she in general was focusing on Streptococcus, which is a sort of dangerous or damaging or infectious disease that's well worth fighting. Generally asking and answering questions about why these strains were so aggressive, how does antibiotic resistance come up, you know, how can we find like treatments against them? So not at all going about looking for like wanting to create a genome editor or something like that, right? So in 2006, she was studying the genome of Streptococcus pyogenes and found and noticed these distinctive series of sequences. So it's in black here. And these were known, it's not like she was the first person to notice these. Since the 80s, they'd kind of been noticed and people were increasingly studying them in the 90s and the 2000s. So they're clustered, as you perhaps have heard of. CRISPR stands for clustered together in the genome, regularly interspersed. So we'd see them and then there'd be something else and we'd see them again, and see something else and we'd see them again. Short repeats. And they're palindromic. That turned out to kind of not really matter all that much perhaps, or maybe we haven't figured out why that matters. They're not necessarily fully true palindromes. So like even just in the English language, a palindrome can be read the same forwards and backwards, right? And these sequences are sort of like that. They're maybe not necessarily perfect palindromes. But these were noticed and people were trying to figure out what they did, and she found them in the things she was studying. So in bacteria, to back up or to kind of take a look at this, how this works in bacteria, bacteria want to have an immune system against viruses. So they use this CRISPR DNA and some other machinery here as sort of an immune system to fight off viruses they've encountered before. So these CRISPR sequences, CRISPR is just referring to really the sequences, are interspersed with what was determined to be little snippets of viral DNA, samples of DNA of viruses that had previously infected this bacteria or other bacteria that had been passed on to this bacteria. And little snippets of the viral genome was sort of inserted into the bacteria's own genome, and it was keeping track of the viruses that it had fought off before. And it can grab those, insert them into this sort of CRISPR-y area of its genome, and use those to fight off viruses that come back later. So how does that work? So, aside from the fact that it collects samples of previous infections, viruses, it's able to put those to use and fight off new infections and viruses. So the process here, and these images are from the sort of Nobel Prize committee themselves explaining this. So they, once you've got the DNA packaged up into the bacteria's genome, it can take that DNA and sort of read it out, type it out as a new strand of RNA, CRISPR RNA, and sort of do that and churn out lots of it. From there, this other RNA comes along, and this is sort of part of what Charpentier figured out, is that there's these key tracer RNAs or trans-activating CRISPR RNAs, little critical pieces of RNA that would go and find those CRISPR sequences, right, the black sequences that are all over, would bind to it. When that happened, along would come, there's where it's binding, and along would come a protein, Cas9. And Cas just refers to CRISPR-associated protein, right? So like, as people are figuring this out, there was Cas, there's this CRISPR-associated protein, and then there was another one, so they're like, well, we'll call that Cas2, Cas3, 4, 5, right? There's all these proteins, and people are figuring out what they do. So this one was the 9th one, CRISPR-associated protein 9, and that would go and bind, and when that happened, this other character would come along, RNase, that would snip the CRISPR RNA on either side and release it, and you'd end up with this sort of genetic scissors. And so you'd have this little package of a little protein, Cas9, and Cas9 is sort of the genetic scissors in this scenario. It would have this tracer RNA still in there, but it would have the CRISPR RNA, including sort of the sequence that, these colored sequences came from the viruses that had invaded previously, right? So it's going around with this viral sequence, knowing that if it encounters this sequence, that's viral DNA that it wants to fight off, right? And so all these different sequences can float around. And if at some point, the same virus comes along and tries to infect this bacteria, it can notice and bind to that sequence, the matching sequence that it originally came from. And then Cas9, when it binds like that, Cas9, the protein, can snip the viral DNA and prevent the virus from infecting the bacteria. So it's a relatively straightforward, simple system. There's really not that many moving parts involved. So this is what Charpentier figured out, published just back in 2011, sort of published this. And at that point realized that she kind of was onto something. There's a powerful little system. And she wanted, as a microbiologist and geneticist, to collaborate with someone who is really competent and confident in RNA. So that's Jennifer Doudna. So she's a biochemist, educated at Harvard and was at Berkeley and worked with RNA quite a lot. And so Charpentier really sought her out, recruited her, and said, hey, we should collaborate. I want to talk to you about this system. So they literally just, less than 10 years ago, got together at a conference, went out for coffee, walked around town together talking about these topics, and thought of some things that they could do. And they realized that they could program, so to speak, this system. So if we go back here, right? Like if this stretch of DNA is really what's tagging what could be cut, they're like, it's probably pretty easy to design our own sequences and have it go attack and cut whatever we want, whatever DNA sequence we want, right? So just, like a year later, 2012, 8 years ago, they published this, this article about programming these little tracer RNAs in Cas9 and such. And so the system is pretty much the same. We very much these days have the ability to sort of print out, so to speak, or create whatever little RNA sequence we want. So if you design it just right to look like the sort of tracer RNA and the guide RNA, just like bacteria use, then the Cas9 protein, you can borrow that from bacteria, put them together, and then it can go target and cut whatever little sequence we want. Whatever sequence you put here is what it'll go cut. So it's very simple, straightforward little system. There's a couple things you can do with it, and so that's what they did. They sort of created this, put it together, and the published, the paper is sort of showing that it worked, and it did. And so there's a couple ways that this can be used. The simplest way is that you send it in and it cuts at a spot, and then you let the cellular machinery take over, and when it repairs this cut, it probably makes a mistake. It probably doesn't just reseal it back up exactly the way it was. There's probably typos or some extra in there or something lost, such that whatever gene this was or wherever it was that you were disrupting or that you were attacking is disrupted. So maybe you turned off sort of like a knockout technique. You've taken a gene that was there, you knocked it out, it's not working anymore. So that's the most straightforward, right? And nothing else is required than sending in the scissors to the right spot. The other thing that you can do, and since then people have sort of built onto this and can do all kinds of things, but the next simplest thing you can do is if you want to insert something, you can cut this gene and then you can send in just the right DNA sequence that's going to match up. And if you flood the cell with a lot of those, so you flood the cell with scissors and you flood the cell with this like insert that you want them to take up, then the cell will sort of glue that in as it's fixing this cut, and you've now inserted something, right? And people have built on this now. There's more advanced setups where even like tethered to this scissors protein is like a gluing protein. It's copy paste. You can kind of build up this machinery to be more complicated. But at its core, this is how it works. It's pretty straightforward. And so this immediately took hold, right? So people were studying CRISPR 10 years ago. But within 8 years, right, it's extremely popular. Tons of people are studying it. Their initial article has been cited thousands of times. And now already 9 years later, they've got the Nobel Prize, right? So they are, tidbit, are the first women to share a Nobel in the sciences without a male co-laureate. And prior to these two, there had only been 5 female chemistry Nobel laureates. So what's the big deal with CRISPR? We've kind of already alluded to it. Super easy to use, and I won't read through these quotes, but basically the technique, once people have figured out how the technique works, it's really straightforward to the extent that people are like, yeah, a high school student could do this, right? Whereas previous techniques were very complicated and cumbersome and took a lot of expertise and equipment. It's very customizable. You just type in a different sequence into your RNA synthesizer, and you can send it wherever you want. It's very fast. So back in the day, it took a couple of years to get a certain kind of thing done. Now you can get it done in a month or 2. It's very easy to scale up and to do sort of genome-wide manipulations by sending in lots of sequences. Because of all of these things and other reasons, it's very cheap. So it's fantastic, right? Very popular. Just to very quickly mention some of the things just in the last 8 years people are doing CRISPR, using CRISPR-Cas9 to do. Lots of medical treatment, so people are doing cancer, using it to fight cancer, immunotherapy. Sickle cell anemia and beta thalassemia have been targeted with CRISPR-Cas9. Just this year up at OHSU, this is kind of a big milestone. So normally when people use CRISPR-Cas9, even to do like human medical treatment, you take some sort of tissue or cells out of the patient, you edit them, get it just right, and then you put them back into the patient. For the first time, just back in March, up at OHSU at the KCI Institute, they sent CRISPR into like the human body, right? The eye is nice because it's pretty contained. You're wanting to work on retinal cells. So for the very first time, they sent sort of CRISPR in, and within the human body, they did the edits. And you got to be really confident that what you're doing is going to work, right? So because once you've done it, you've done it. So anyway, that's pretty cool. Big news items. So aside from treating people, right, we can attack viruses, and people have done work like if we were bold enough to do this in people, we could, but they've already engineered mice. They've gone in and sent the CRISPR into mice and edited them such that they're resistant to HIV. You could use CRISPR to attack the viruses themselves, like HPV. You can create viruses, bacteriophages, that go around and attack the bacteria for you. People are doing it to edit crops in various ways. They've edited tomatoes to just branch more and produce more fruit per vine. And CRISPR, importantly, and some other related techniques are not actually GMOs. It's a different technique. GMO techniques, sort of the way they're defined and legally the way they're on the books, it's kind of messier. You're sending in maybe other genes and such. Gene edited, it's more precise. You're going in and sort of carefully adjusting things. So in some ways, it's there's maybe a lower barrier for regulation, and it's separate from existing regulation about GMOs. People, you know, you can Google these things on your own, but you can eradicate pests. People are talking about de-extincting things that like are human-caused extinctions of populations of animals that we could de-extinct, or maybe they're on the verge of extinction. But another really important one to highlight is it's a super important technique just for doing basic science, right? Previously, it's just a really powerful tool that lets us do really powerful science. So it's kind of like if the way things before we were using a screwdriver, you know, we now have a drill, and it, you know, it does the same thing. It's not necessarily something we couldn't do before, but it's much more powerful, much faster. So it enables, it's just a tool that enables us to do a lot of other things. So, you know, I won't take too much time here, but much like Kamal had take-homes, you know, interdisciplinary research and interdisciplinary collaborations, or just collaborations in general, tend to be really powerful in science. And basic science research is super valuable, right? In the 80s, when people are looking at these weird palindromic sequences in bacteria, right? It's like, why should we fund that? You could argue, like, why do we need to fund studying these sequences in bacteria? At most, you're going to come up with some sort of new antibiotic or something from that, right? Like, no one could have predicted that it would lead to this super powerful tool to do lots of other things. So, aside from some recommended listening and reading, I should stop talking and see if we have questions.
So there was the news story about the babies that were Was that to make them HIV resistant? I thought.
I'm gonna also tap Kamal off and on if he wants to ever speak up, because of the microbiology side of things, he's followed this as much or more than me, I think. They were to be, was it HIV resistant? Yeah. Yeah, similar to the mice mentioned,
Okay.
Right? So in 2018, there was this sort of big hubbub about rogue scientists editing human embryos. So various people have edited human cells, human embryos, but nobody has implanted, other than him perhaps, or other people that we don't know about, maybe, implanted them, carried them to term, or them grow up with the children. Yeah.
So do you think that one day of like transgenic, gene knockout, anti-scans, all those will be phased out and replaced with CRISPR?
Well, I didn't mention it here. Obviously, CRISPR has drawbacks, right? There's issues with like being on target and maybe too much off-site editing and things like that. I imagine there will always be a mix of other techniques in play. You know, just like screwdrivers still exist, even though we have power drills. But maybe usually we grab the power drill. And it's fine if there's not a ton of questions, because we don't want to leave time for black holes.
Can you talk a little bit more about the cutting process? Which chemical bond?
Oh yeah, the cutting.
That's not great.
Yeah, so there's a DNA, you know, phosphate backbone. DNA has a phosphate sugar backbone. Look all the way back, and the scissors here represent parts of the Cas9 protein, particular little sites that, when things are positioned just right, they attack the ribose phosphate backbone in DNA, and so you cut that apart and then just let it go, and you know, now it's cut apart. Yeah, but there's you know, it's features of this Cas9 protein that lead to this Cas9. Any others?
Can we, or do we do this in our labs here?
I don't know. We could. I think we could. I don't think
There are kits that are coming available that are probably not too expensive. We don't have them, or we don't have the tools yet, but it is something that we could probably at some point do.
Yeah, like you said, high school students, right? I want to be sure to leave time for black holes, but I'll be here at the end if you guys
All right.
have a question.
Okay. Well, we decided that the other two prizes had some logical connection, so they should go back to back. But there really wasn't much of that with the physics prize. So black holes are really different than anything we know about on Earth. But that's what the 2020 Nobel Prize was awarded for. So I like these images they have at the Nobel Prize site. this just shows someone contemplating a book, sitting under a tree reading, and maybe blissfully unaware that there's a supermassive black hole at the center of the galaxy right up above their head. Half of the prize this year went for the discovery of that supermassive object at the center of our galaxy, and the other half went to a mathematician and physicist named Roger Penrose, who back in the 60s convinced everyone to take black holes seriously. So Roger Penrose, over here on the left, he got half the prize for his contributions to the theory behind black holes. And the other two prize winners, Reinhard Gerner and Andrea Ghez, they got their Nobel Prizes for these meticulous studies they did of the stars at the center of our galaxy that basically leave us no conclusion other than the thing at the middle is a huge black hole. So a lot of the prizes in physics recently have had something to do with black holes or general relativity. And rather than focus too much on the individuals, I wanted to focus more on kind of the story that they're a part of. So the specific prizes were given to Roger Penrose for the discovery that black hole formation is a robust prediction of the theory of general relativity. So we hear about black holes in the movies, if not the news, a lot these days. But for a long time, people thought, well, this is just some kind of curiosity, some weird solution to a mathematical equation that doesn't have any relevance. And what Roger Penrose did was show that actually, if you're going to accept any of general relativity, Einstein's theory that I'll talk about in a moment, you have to accept black holes. They're a part of it. They come with everything else that's part of the theory. And Genzel and Ghez, as I said, they basically made very meticulous measurements of the motions of stars and showed that something that is either a black hole or something even weirder than a black hole has to exist at the center of our galaxy. So I went through the list of Nobel Prizes and looked at all of the ones that had something to do with general relativity. So when I say it's part of an ongoing story, you can go backwards in time. The last 4 years have been great for general relativity. We have black holes this year. Last year, it was Big Bang cosmology and applying general relativity to understand how the universe as a whole evolves. 2 years before that, we had the discovery of gravitational waves with LIGO. And going back further, there were some discoveries in cosmology. At the time, these cosmic X-ray sources were recognized. People weren't sure they were black holes, but they were really weird things that now turn out to be black holes. But you can see there's a huge gap between 1978 and 1921. 1921 was actually when Albert Einstein got his prize. And I was amused when I read his prize description because it was just for services to theoretical physics. Like he did so many different things, they didn't know which one to really recognize with the prize. So he got a sort of a lifetime achievement award while he was still in his 30s. So, so let's go back to Einstein and see a little bit about what general relativity is. Okay, so this is the challenge you always run into if you're trying to explain general relativity. He starts out talking about the rubber sheet over here on the left. The student says, well, what's the sheet? Why is it bending? And so if you really want to understand relativity, you have to resort to the mathematical description, but that's kind of hard to hold people's interest with. So I want to present some of the important results to you and give you an idea of what's going on in this theory of general relativity, but we don't have time to go into all the details. Okay, so this is the man, I did get the date wrong. It was actually 1915 when Einstein published his theory of general relativity. But the theory was very different than anything that had been seen before. It forced us to completely revise how we think about time and distance. So when something is moving quickly, right, the rate at which its clocks move slow down. And when something is moving quickly, someone in a spaceship that's moving quickly and you will disagree about how far apart things are. So according to Einstein's theory, time and distance are relative. There's some things that are not, which is even weirder. So So one of the centerpieces of his theories is that the speed of light is not relative. It's the same for everybody. If I am flying towards you at half the speed of light, and I shine a flashlight beam at you, there's no extra speed added to that light. It still reaches you at the speed of light. And if I'm moving away from you at 99% the speed of light, shine a laser pointer at you, it still reaches you traveling at the speed of light. So that's a very, very strange fact about our universe. And Einstein's theories of special relativity and general relativity are basically our only way of explaining that consequence, that fact. And so one of the things he did first in 1905 was to develop this theory of special relativity, which just deals with things that are moving around in empty space. And there was no information at all about gravity in there. And so in 1915, 10 years later, he published his theory of general relativity that incorporated gravity into the whole mix. And the way he did that was to explain how gravity warps space and time. And also, gravity is actually more like light than you might imagine. Gravity takes time to travel through space. It travels at the speed of light. The measurements by LIGO, they got the Nobel Prize a few years ago, confirmed that. The difference between the speed of light and the speed of gravity waves is so small that we weren't able to measure it. And so gravity is more than just the force that pulls us to the ground. There's a lot of unusual stuff going on. And in 1919, that's actually when Einstein became a celebrity. So when he published his theory of general relativity, he explained two things that no one else had been able to explain, but he also made a prediction about what should happen. So we learn about planets moving in circular orbits or elliptical orbits in classical physics, but actually that's not the case for something like Mercury. The closer you are to a star, the less accurate Newtonian physics tells you is for predicting the orbits. So Mercury's orbit does something called precession. It moves in an ellipse, but the ellipse moves around. And so if you look at its orbits over thousands of years, it actually looks more like the pattern you would draw with a spirograph than some ellipse that just has the sun at one focus. And so Einstein's theory explained that, and Newton's did not. And so he explained one confusing fact that already existed. Another fact that he was able to explain is why light coming from the sun, from certain transitions in atoms, looks redder than light coming from the same atoms on Earth. So gravity has an effect on the wavelength of light. But the prediction that no one had tested yet was that the sun should bend light that's passing by it, and should bend it by a very precise amount. So in 1919, someone went out to a very remote island off the coast of Africa and took photographs of a total eclipse of the sun. And the reason it had to be a total eclipse is because you wanted to see where the stars were in the background. They're kind of hard to see in this image, but you get an image of where the stars are when the sun is between you and the stars. And you can compare that to where the stars are when the sun is not there, when you take it on a clear night. And you can measure the shift in the position of the stars. And that shift matched Einstein's prediction of general relativity almost perfectly. And there was a neat string of recent measurements when we had our eclipse in Oregon in 2017, where they repeated these measurements and confirmed yet again how the sun's gravity bends starlight. So these are the kind of things we have to confront with general relativity. But there's even stranger stuff. So about 6 weeks after Einstein published his mathematical description of how gravity and space and time all fit together, a mathematician named Carl Schwarzschild said, "Hey, I found a solution to those field equations." Right? This is just to give you a taste of what the equations that describe space-time actually look like. It's something called the space-time metric. And what it tells us is the distance between nearby points, or the time interval between events that happen. And so the t's and the r's and the angles, those are our usual coordinates. But Schwarzschild's solution has some interesting properties to it. So one thing is that it tells us exactly how matter is changing the rate at which clocks flow or the distance you measure with a ruler, but that was already part of Einstein's theory. This solution has two special points. Right? So what it's describing is a spherical distribution of mass, and R is how far you are from the center of that sphere. And if you look at the equation and do some algebra, you find there's two places where something very bizarre happens. So one, if R is 0, if you're right on top of this point mass, some things in the equations become infinite. Things like how far it is from the center of the circle to the edge, or how long it takes to get from point A to point B. So that's what we mean when we talk about a singularity. It's a place where space and time don't behave in conventional ways, in ways that make any sense at all. The other interesting value is what happens when R takes on this value twice Newton's gravitational constant times the mass of the thing divided by the speed of light. And some things become 0 there, or infinite again, but this is a little different. It's something called an event horizon. People realize, well, if you had a rock or a spaceship or a person that was moving towards the center of this thing, and they got past that point, they could never get out, no matter how hard they tried, no matter how fast they were trying to go. If they shined their flashlight backwards, it would never come out. Right? So this is actually a mathematical description of what we mean by a black hole. And Einstein didn't like it. He thought, well, that can't exist in the real world. But if you just plug in the numbers and see when you might expect to see these odd effects, for the sun, you would have to take all of the mass of the sun and compress it down into a distance smaller than 3 kilometers, smaller than 2 miles in radius to see these effects. For the Earth, you would have to take the whole thing and compress it down to a sphere that's less than a centimeter in diameter. Right? So we aren't going to notice these effects because our matter is so spread out. And so it's very hard to test this prediction, especially back in 1919 or 1915. So people were aware of this solution. It became more and less popular over time. Robert Oppenheimer studied what happens when you have a bunch of stellar matter that's evolving in time and it's collapsing. And he found that actually you could start out with something that was a star, and as it burned up its fuel, it would collapse and it would go into this Schwarzschild solution. But some other people said, well, you're assuming spherical symmetry. Nothing in the universe is that perfect. And we don't actually know what happens if you start out with something that's not a perfect sphere. And so people thought, well, maybe it's just a mathematical anomaly. But in the 60s, we discovered objects called quasars. So these are objects in distant space. They're very compact as far as things in space go, much smaller than the size of our galaxy, but giving off more than 2,000 times as much light as all of the stars in our galaxy combined. And so then interest in black holes started to pick up again because it was the only thing that anyone had any idea about that could give up that much energy from that small of a volume. And so that was the state of things when Roger Penrose came onto the scene. What he did was develop some new mathematical tools for understanding the predictions of Einstein's theory. So he wanted to get away from this requirement of perfectly symmetrical solutions to Einstein's equations and see if he could describe what happens in general. Are black holes real? Are they a robust prediction of general relativity? Or is this Schwarzschild solution just one perfect lucky coincidence of the equations? And so he developed new ways of analyzing what happens in a collapsing star or when things get too close. This is an example of something called a Penrose diagram. But what he found was that he could make topological arguments about black holes and collapsing stars that don't depend on the shape at all. You can start with any kind of blob you want. As long as the mass is big enough and the radius is small enough, it's going to make this thing, something called a singularity. So he has a mathematical proof of something called the singularity theorem. And he developed diagrams that let you do incredibly complex calculations, but by drawing pictures like this instead of working out the math. And so lots of people could do the calculations and study many different systems. And basically what this shows is that if you take any part of general relativity seriously, you have to take black holes seriously too. Black holes are a prediction of general relativity, and they have singularities at the center where space and time cease to exist. And we have to confront that if we're going to push this theory to its limits. And people have called it the most important contribution to general relativity since Einstein. It's what set Stephen Hawking on a lot of his investigations of black holes, and other scientists you may have heard about. Penrose really got the ball rolling with that. So that's why he was recognized. The other thing we want to do, though, is understand these black holes. And Penrose says you can't look inside. Right? There's this event horizon, nothing can come out. If you go in, you can't come back out. How do you study it? Well, you look at how it affects everything that's outside of the black hole. So this is a picture of Cygnus X1. It's a really bright X-ray source in the sky. And what people found is it's giving off so much radiation, and it has a partner star that's orbiting it. And we know that the thing giving off the radiation is smaller than the orbit of that partner star. So basically, we have an object whose only possible explanation that's not more exotic than a black hole is a black hole. The other two scientists who won the Nobel Prize, Wenzel and Yes, won for their astronomical observations. So they developed new techniques for looking at the center of our galaxy. They were trying to map the orbits of stars around the center of the galaxy to figure out what's there. And it's really hard. There's a lot of interstellar dust, and the center of the galaxy is really bright. And so if you imagine trying to look there and track the orbit of one star out of all the billions in our galaxy concentrated in the center, it's very difficult. So they found that by observing in infrared and radio ranges, you could watch these stars. They developed new techniques for imaging, something called speckle imaging, where you take thousands of pictures of what you want to see and put them together to get a high-resolution image from a bunch of low-resolution pictures. And they used adaptive optics. This picture on the left shows the Keck Observatory in Hawaii, and they fire a laser up into the outermost reaches of the Earth's atmosphere and excite a bunch of sodium atoms up there and make an artificial star. And then they have mirrors that can change their shape at the snap of your fingers. And so they use that known guide star that they're creating artificially, change the shape of the mirrors in the telescopes to compensate for what's happening in the Earth's atmosphere, and get incredibly precise pictures of what's happening at the center of our galaxy. So these technical developments were a big part of what they won the prize for. When they did that, I know I'm running short on time. So when they did that, they were able to map the orbits of stars at the center of our galaxy. If you want to know how patient some of these scientists are, they collected data for 26 years to track these stars going around the center of the galaxy. And they found one object, Sagittarius A star, that is the center of our galaxy. Everything else in the galaxy orbits it. And they found one star in particular that comes within 17 light hours of the center. So that's about 4 times as far so Pluto is about 4 light hours from the sun. So this star is approaching almost that close to the center of the galaxy. And by using Kepler's laws, these laws for how things orbit, and Einstein's corrections, they're able to figure out how big this thing has to be. How big is Sagittarius A? And so what they find is that it basically is some very compact object, fits into less volume than our solar system, but it has a mass that is 4 million times the sun's mass. And the way they learn that is pretty interesting, but I think I not have time for that. Right? And so this is it. This is the picture of the monster at the center of our galaxy. 400 million solar masses combined, condensed into this small volume. Everything is orbiting it, and really there's no other explanation for what it could be other than a black hole. So you may remember the Event Horizon Telescope a couple years ago gave us our first actual image of a black hole. People are applying the same techniques to Sagittarius A at the center of our galaxy. So we should soon actually get photographs of the black hole at the center of our galaxy and learn more about it that way. But the prize was for figuring out what that thing is with these indirect methods. What comes next? Just from Penrose, we know that singularities are a prediction of general relativity, but general relativity breaks down there. And so the big open question in physics is what comes next? How do you go beyond general relativity? How do you combine it with quantum mechanics to understand even more about this universe that we live in? So I know it's class time for some people. Thank you for listening. But if you have questions, I can answer a couple as we're taking stuff down here. Thanks everyone for joining.
Thanks, Jesse.
Zoom.
Thanks for organizing.
Oh,
Thank you.
yeah, you're welcome.